Hi everybody, I'm Colin Fogarty and this is your Biology SOL Review. Now I'd like to take some time before I begin this video to just apologize because I'm going to be reviewing things out of order. So if you just go to the comments below, you can check for the timestamp for when I talk about different parts of the curriculum. You can just select that timestamp and then it'll take you to that part in the video. But without further ado, let's begin, and we're going to have to begin with the scientific method, that thing that you've been reviewing since second grade. So every experiment has to have a hypothesis, and most hypotheses will follow the if-then statement format. If I throw this mouse, then I'm going to break the window. But they can also basically just be any statement that guesses what's going to happen. Throwing my mouse will break the window. But every good experiment has to have an IV and a DV. And an independent variable, or IB, is the thing you're going to change. And the dependent variable, or the DV, is the thing that's going to become changed as a result of the IB. But a good experiment also has to have constants and a control. Now, a constant is something that stays same throughout the experiment, whereas a control is the base product or thing that you do not change in any way so that you can gather accurate data that you can measure against your other dependent variables. Now, you have to also gather data in experiments. You can gather either qualitative or quantitative data. Now, qualitative observations are going to be what you hear, or what you see, or maybe what you taste, anything that you can do with the five senses. But quantitative observations are going to be anything that you can actually measure. So now let's touch on the parts of a microscope. So each microscope has a base, and with that base there's a light source. And so extending up from that base is the arm on which the stage, the revolving nose piece, and the ocular part of the microscope are. So the stage is going to hold your specimen or your sample down with a stage clip, and you're going to use that revolving nose piece to see the sample through the ocular part. Now, most eyepieces, like the regular ocular thing that you're going to be looking through, are going to have a 10 times magnification, and the revolving nose piece are going to have magnifications of 4, 10, and 40, and so you multiply that eyepiece against the zoom that you have on the revolving nose piece to get your net zoom. So if I have 10 in my ocular but 4 in my nose piece, that gets me a net power of 40. Okay, let's talk about classification. So you need to remember the order, which is kingdom, phylum, class, order, family, genus, species. And I don't really have any good mnemonic devices to give you. I would just say drill it in your head that it's kingdom, phylum, class, order, family, genus, species. Now, you really only need to know the 7 kingdoms, which are eubacteria, archaebacteria, animalia, plantae, fungi, and protista. And don't forget the naming system that we use for species, which is called binomial nomenclature, and it has 2 parts, hence the binomial. The genus and then the species. Now, if the SOL is going to require you to read a dichotomous key, then make sure you look at the organism and then just follow it based on what you see. You don't have to overthink it, and it's going to be very clear for how you follow a dichotomous key on the SOL. Let's talk about ecology. So for this, I'm going to actually draw us a food chain. So at the bottom, we have organism 1, and that's going to feed organism 2, and that's going to feed organism 3, and that's going to feed organism 4. Let's just say that organism 4 is a hawk, this is a snake, a bunny, and then grass. So in this food chain, because it's just one single way, we're measuring where the energy is going. So the energy starts with the grass, and the grass is going to be our producer. And our bunny is going to be our primary consumer, because they're first. So primary, I'm just going to put con. Now, what is our snake? Our snake is our secondary consumer, because they're going to be consuming something, but they're second on the chain. So they're a secondary consumer. And lastly, we have our hawk. Now, our hawk is going to be a tertiary consumer. And you can remember that tertiary means third in the order because of the T. T for tertiary. Now, let's also examine some relationships. Predator, prey. The hawk is the predator, and it preys on the snake. But the snake and the bunny also have a predator-prey relationship. Predator-prey. So prey can also be, so what is one predator to another prey can also be the prey to another predator. Now, we can take this food chain and we can elaborate on it. So we could have, say, a bird here. And that's going to eat the grass. And we could also have, say, oh, I don't know, a mouse. And the mouse is going to eat the grass too. So our hawk could eat that mouse. And maybe that snake eats this bird, right? So the bird and the mouse are still going to be primary consumers, but they're also going to be the prey for the snake and the hawk. Now we also have to talk about decomposers and scavengers. Scavengers are something like a vulture. Now if a mouse were to just die and then a vulture were flying around, they might come down and eat that mouse because it's dead, but they're not going to actively go out and hunt stuff. A decomposer, however, might be a fungi. And when that mouse dies, that fungi is going to just basically cover that mouse, and it's going to break it down, and it's going to help make more fertile soil. Now we can also talk about symbiosis while we're here. So allow me to erase this. So symbiosis is any relationship between two animals that involves being in or on or around. So we have three types of symbiotic relationships: mutualism, parasitism, and commensalism. So let's just draw out what the faces would look like. Parasitism is one happy face because the parasite benefits off of the other animal, but the other animal, oh, he's sad because he's being taken advantage of by the parasite. So that's parasitism. We'll just label that with a P. Mutualism, well, have you ever heard the expression the feeling is mutual? Well, that means you feel the same way. So somebody's happy. Oh, and look who else is happy. Both animals are happy. Both animals get something out of that relationship. And the third one is commensalism. And this is probably the hardest for me to remember. Commensalism is basically you have something good, but you also have somebody that just doesn't care. It's like, yeah, dude, I don't really care. Do what you need to do. So we have a happy face, and we have a meh, okay, whatever. All right, so here are just some more terms you need to know about ecology. So carrying capacity is how much of a certain population that a environment can sustain. How many of Jack Lewis's can my biology class contain? Well, it's really only one. That's the carrying capacity. Any more and we can't sustain it. Now, carrying capacity can also affect competition based on how much of a certain population you have, as two species are going to compete over food. Back with that food web example I provided, both the hawk and the snake are competing over eating that rabbit because the hawk can also eat the rabbit, if that were the scenario that we had. Now we also have to talk about succession. So if I have a forest and that forest just totally burns down, and then a new environment takes its place, that's called succession. Like succession after the crown, or just basically coming after something. Now there's primary and secondary succession. With that forest fire example, that would be secondary succession because we already had an environment there before. But if there's just an environment that just comes out of nowhere, say like a glacier retreated, then that would be a primary succession. So now we also have to talk about things related to ecology that are the cycles. So that's the water cycle, the carbon cycle, and the nitrogen cycle. I hope you all know what the water cycle is, but we can review it. Basically, water goes up into the sky through evaporation, becomes water vapor through condensation, it gets put back onto land through precipitation. But don't forget about transpiration, because plants can absorb water and then give off water vapor. Now the carbon cycle is different, obviously, because it involves carbon. And it involves plants using that carbon for photosynthesis, and then animals eat those plants, and that when they breathe out, like when we humans, we exhale, we put that carbon back into the atmosphere, but we can also die, and then we become fossil fuels, and then those fossil fuels are burned off, which release carbon back out into the sky. And so lastly, we have the nitrogen cycle. So first we have to understand that we can't actually use nitrogen in its base form, which is N2. So we have to fix it to be usable in a process called, guess what? Nitrogen fixation. So once nitrogen reaches the soil, either by like an organism dying or through precipitation, bacteria are going to help it bond with hydrogen to form NH4, aka ammonia. But we still don't really want to use ammonia. So bacteria are going to then transform that NH4 into nitrite, which is called NO2. But after that, it's going to become nitrate, which is NO3, so we plants and animals can finally use it. But when we die, we can put that nitrogen back into the soil. But how can we put nitrogen back into the atmosphere? Well, we can do that through a process called denitrification, which is when bacteria basically break it down and put it back into N2 so we can put it back into the atmosphere. So now we've covered ecology and classification. Let's touch on viruses and bacteria. So a virus is a non-cellular structure that is covered in a protein, and then it infects and reproduces in two ways: the lytic and lysogenic cycle. So the lytic cycle involves a virus penetrating a cell, and then it infects it by entering it. This is and then in a process called biosynthesis, the virus uses the cell's reproduction mechanisms to reproduce those viral proteins. And so once they have enough viral proteins in a process called maturation, the cell is going to burst in a process called lysis, and then all those viral proteins that have been reproduced are going to infect their own cells. And then imagine that process over and over and over again. We're going to eventually have tons of viruses. But we can also do it through a process called the lysogenic cycle. So the lysogenic cycle is similar. We have a virus that attaches and then penetrates into a cell. But instead of using biosynthesis, it actually molds its own viral proteins into the DNA of the cell. And so when the cell divides, it's going to just keep on dividing this infected cell. And then bacteria, basically what you need to know about bacteria is that they don't have a nucleus. And that's pretty much the most important thing. They have a cell membrane, but they don't have a nucleus, which is the biggest difference between a prokaryote and then a eukaryote. Prokaryotes do not have cell nucleus, or nuclei I should say, but eukaryotes do. All right, so now we're going to start moving towards smaller and smaller units of life. So now we have to discuss what photosynthesis and respiration are. So photosynthesis and respiration kind of work in tandem with each other. Photosynthesis requires sunlight, water, and carbon, and results in oxygen and glucose. So the equation for this is 6H2O plus 6CO2 equals H6C12O6 plus O6, which is glucose and oxygen. So that respiration, which is the process that follows photosynthesis, is going to use something called ATP as its energy, and the equation is basically the reverse of the photosynthesis. So you put the glucose and the oxygen in, and you get water and carbon out. So what does this cycle actually look like? Well, first you have sunlight, and then the chloroplasts are going to take it in, and they're going to use the water and the carbon to create glucose and oxygen. However, our mitochondria, powerhouse of the cell, they're going to take in those products and they're going to use them as reactants with ATP, and they're going to create carbon and water. Think of it as how we humans breathe, right? We take in carbon and then we put out oxygen. But when we're working out and we're breathing, you're putting out the carbon, we're taking in the oxygen, we create water because we perspire, and that's how you can kind of think of photosynthesis and respiration. All right, let's move into cells. So first we have cell parts for a eukaryotic cell, or any cell that is part of an organism that's not bacteria. Because remember how I mentioned that prokaryotes do not have a nucleus, whereas eukaryotes have a nucleus. So first we have, obviously, a nucleus, which is the command center, with something called a nucleolus, which is kind of like a addition to that command center. And then we have a nuclear membrane that surrounds it. And then our DNA and our chromatin are going to be inside of that nucleus. Now, surrounding the cell is going to be something called a cell membrane, and that's going to determine what is going to get in and what is not going to get in. And plant cells are even going to have something called a cell wall that's going to really help protect it. And so filling our cell are going to be two things: cytoplasm and a cytoskeleton. The cytoplasm is just kind of like a jelly that just surrounds everything, and then a cytoskeleton is going to help provide with us some structure. Now, there's an endoplasmic reticulum, and that's going to be close to our nucleus, and we're going to call it just ER for short. Now, there's two types of ER. There's smooth ER, and then there's rough ER. And the way you can tell the difference is rough ER is going to have dots on it, and those dots are actually going to be ribosomes. And so what are ribosomes? Well, ribosomes are going to help us manufacture proteins. And when we talk about DNA replication and translation, all that kind of stuff later on, you'll see the real importance of having ribosomes. So our vacuoles are going to store our waste, and then lysosomes are going to help us digest or break down that waste with the enzymes that they carry within them. And then lastly, we have a Golgi body, and what their purpose is they're going to move things around the cell, and you can think of a Golgi body as like a mail service. So now that I mentioned a cell membrane, allow me to elaborate on that structure. So cell membrane is a semi-permeable structure, which means that some substances can get in and other substances cannot. And it's going to be made up of phospholipids and proteins. Now, on the top of the membrane, we're going to have heads, which are going to be hydrophilic, which means they want water, while the tails, or the parts that are going to face into the cell, are going to be hydrophobic, which means they don't like water. Remember, like phobia or philia. And then the cell membrane is going to play a huge role in transportation, especially with things called channel proteins, which moves us into cell transport. How wonderful and how convenient. It's not like I planned that in any way. Now there are three types of solutions: hypertonic, hypotonic, and isotonic. So water is going to move out in a hypertonic and it's going to shrink. Think of it like when you're hyper and you got to get all your energy out. A cell is so hyper, it has too much water, it's got to get that water out of there. On the other hand, you have water that's going to move into a hypotonic solution, and eventually that hypotonic solution might actually burst. So think of it as a very hungry hippo. Hungry hippo, hypotonic. And lastly, isotonic is neutral, and water isn't going to necessarily be moving in or out. It's just going to kind of be very neutral in how it treats everything. And now we can talk about our types of transport, which are active transport, facilitated diffusion, exocytosis, and endocytosis. So active transport involves ATP actually carrying molecules across the membrane, while facilitated diffusion is basically those molecules, they just move across. They're not going to require that ATP to move across the semipermeable membrane. So now we have exocytosis, and that involves a cell basically like pushing a molecule out or a substance out until it kind of just, well, it just gets burst out. Whereas endocytosis is different because a cell or membrane is going to basically surround something, and it's going to pull it in until it kind of becomes absorbed by the cell. So on the note of like literal cell transport, single cell organisms can use three ways to kind of transport themselves or move themselves around, and that's cilia, which are kind of like little hairs, pseudopods or false foot, and those kind of help move them around, or propulsion by a flagellum to move around, and you'll see these in things like bacteria, protists, stuff like that. And so last up to review on cells is the cell cycle. So the cell cycle involves interphase and then meiosis and mitosis. And well, that's what you need to know. And so interphase can be divided up into G1, S, and G2 phases, but you really only need to know the S phase, also known as synthesis, because that's when DNA is going to get replicated. So when we hit mitosis, we're going to begin with prophase, and mitosis is the way that a cell divides. So in prophase, the cell is getting ready to divide, and they're going to move into metaphase, where the chromosomes, they've been lined up in the middle of the cell, and then these things called centrioles are forming on the poles of the cell, so on opposite sides, and they're going to have these things called spindles, and they're going to go out and they're going to touch the chromosomes. So, and that's going to immediately lead us into anaphase, in which those spindles are going to break apart those chromosomes. And then we're going to have telophase, in which basically individual nuclei have begun to form, or membranes have begun to form around those chromosomes that have been pulled apart, and the cell is basically about to split, but it has not split yet. And then obviously the cell splits, but that's after mitosis is done. Now, mitosis is going to take place in somatic cells, and that's basically any cell that's not a sex cell, and it's going to produce diploids, or two sets of DNA from the parents. Meiosis is basically the same, but they're going to have a second round of division in which they're going to produce haploids, or they're going to produce 4 sets of chromosomes, and meiosis is going to take place in the gametes, or the sex cells of an organism. And so, you know, you have all this stuff, but in meiosis, we're going to see something special in which we're going to see crossover, and we're going to see DNA from both the parents kind of mix, and that's going to create diversity in the offspring. Now, keep in mind that the amount of chromosomes a somatic cell has is double that of the gamete. So if my somatic cell has 40 chromosomes, then my sex cell is only going to have 20. So if you have a question on the SOL about that, just remember that kind of math. Okay, so now we can talk about DNA, because I know I've been kind of touching on it here and there. So DNA has a double helix structure, and it was composed of carbohydrates linked by phosphates, and they're going to run in opposite directions. So you might have one running up like this, but on the other end of the double helix, it's going to be running down like that. So it's going to have carbohydrates, they're going to be linked by phosphates, and then they're going to have, they're going to be connected to nitrogen bases. And again, all running in opposite directions. And so the nitrogen bases are going to have guanine, cytosine, thymine, and adenine. And so when you have a nitrogen base, a deoxyribose, or the carbohydrate, and then a phosphate, that's called a nucleotide. So those four nitrogen bases are going to be able to link with each other, but they're only going to be able to link with specific other nitrogen bases. So adenine is only going to be able to connect with thymine, and then cytosine is only going to be able to connect with guanine. And then you can kind of remember this by the sharp letters like A for adenine or T for thymine. Those are the only ones you're going to be able to connect. And then the round letters for like cytosine, C or G for guanine, they're going to be able to connect. Now, DNA is going to be discovered by two researchers. Their last names are Watson and Crick, and that's going to be discovered in 1954. And then there's also going to be a woman you should know. Her name's Rosalind Franklin, and she's going to be able to successfully photograph DNA and kind of help discover that, or help lead other people to discover that it is a double helix structure. But that's not the most important part, because we need to talk about DNA replication. So replication is how DNA copies itself for when it divides, and that's going to happen again in the S phase of interphase, or the synthesis phase. So a helicase is going to come in and it's going to break apart the hydrogen bond that's going to keep those nitrogen bases together, and that hydrogen bond is actually weak, so it can be broken apart by the helicase. And then something called a polymerase is going to move along, and it's going, or move along the DNA strands, and it's going to add the nucleotides to it, and then eventually after that all happens, voila, you have two strands of DNA molecules. So then you have your replicated DNA. You have a double, so when you go into meiosis and mitosis, you have two sets of DNA to split and create new cells. We also have to talk about translation and transcription, and this process involves using DNA to code amino acids. And amino acids are going to help us get proteins, but we're not going to cover that until the macromolecules section. So this occurs in the ribosomes of the cell, remember we mentioned that earlier, and uses RNA. So there are three types of RNA. There's mRNA, tRNA, and rRNA. So an mRNA, or the messenger RNA, it's going to take the genetic code from the nucleus to the ribosomes, and those ribosomes are going to be made of rRNA. So then tRNA is going to carry the info for the amino acids. Then that mRNA is going to form a codon to which the tRNA carries the anticodon, and then those codons and anticodons are always going to match. So now we have to understand complementary DNA or RNA strands. So let's get a 9-base strand. So let's say I have, on my original, I have A, C, G, G, C, A, C, A. And this might not actually be something that you see. So when I translate it, so remember, guanine goes with cytosine, and adenine goes with thymine. We're going to see something particularly special. Adenines are not going to become thymines, they're going to be called uracils. So everywhere that we see an A, we're going to replace it with a U. So U, G, C, C, G, U, U, G, U. And then you can take these three sets, so the three nitrogen bases that you have, and then you can use a codon chart to basically figure out what type of amino acid you're going to code for. And just remember that all amino acids are going to have to start with the amino acid MET, and then they're going to all end with the one that says stop. So now we can talk about genetics. So the father of genetics is a guy named Gregor Mendel, and he really liked playing with pea plants, and that's how he figured out genetics. And so, while that's all nice and dandy, we actually need to learn some vocabulary. So a dominant trait is a trait that will show when paired next to a recessive trait. Dominant traits are like a capital letter, so capital R, little r. And a recessive trait will not show if it's paired with, unless it's paired with another recessive trait. So if I have a little r and a little r, it'll show, but if I have a big R and a little r, the big R is going to show, the dominant trait. Now these pairings can either be homozygous or heterozygous. And just remember that homo is the same and hetero is different. So if I have two big R's, that's a homozygous. But if I have a little r, that's a homozygous recessive. But if I have big R, little r, that's heterozygous. Homo same, hetero different. If you really have to, just remember homosexuals and heterosexuals. That always works for me somehow. Now, sometimes we have to talk about codominance. So let's say I have R and W. So R codes for red and W codes for white, but I get both a capital R and a capital W. Now what's going to happen is we're not going to have red, we're not going to have white, we're going to have pink because these codominants are going to mix together and they're going to create their own phenotype. Wait, what's a phenotype, you ask? Well, phenotype is what's going to show. While as a genotype, its cousin word, if you will, is the genetic code. So I might have RR, and that's going to be my genotype, but what's going to show is red. You wouldn't go up to someone and say, what hair do you have? Well, I have RR hair. You say, well, no, I have red hair. So we can also see mutations. So the types of mutations we can see deletions, and that's when part of DNA gets deleted. We can see substitutions, and that's when DNA might get deleted but then replaced with another one. Then we can also see additions, and that's when just a new thing of DNA is just put in there. And sometimes mutations can be helpful, but other times mutations can be very deadly, especially for infants that are about to get born. Now, we can figure out what kind of phenotype we're going to see based on the genotypes using something called a Punnett square. So let's take this time to illustrate how you can determine what kind of phenotype you might see based on the genotypes using something called a Punnett square. So let's say we're going to code for or determine eyes here. So a big B is going to be blue eyes, and a little b is going to be brown eyes. So we have our parent, our mother, she's going to be heterozygous dominant. So she's going to have a big B and a little b. But the father is going to have brown eyes, so they're going to be little b, little b. So how do we determine genotypes? Well, you just basically add the letters together. So big B, little b, little b, little b. Big B, little b, little b, little b. And you can calculate the ratio. So we have 0 chances of heterozygous dominant. We have 2 chances of homozygous dominant. We have 0 chances. 2 chances of heterozygous dominant and 2 chances of homozygous recessive. But there's another thing we have to cover in genetics, and that's called a pedigree. And a pedigree is how you can trace family genetics. So we're going to do a pretty simple pedigree here. We're going to use squares to illustrate men, and we're going to use circles to illustrate women. So let's see. Now, what can we tell from this pedigree? Well, let's see. We know that this condition, squiggly line condition, is only occurring in 2. So we can assume maybe it's recessive, maybe it's dominant, because the important thing to realize is that who is it only occurring in? Well, it's only occurring in males, so we can assume that it's an X-linked trait. An X-linked trait is going to come from the mother, because you only get one X chromosome from your mother because men have X and Y. So it's very real that because they're only getting the one X chromosome from the mother, they could be getting the recessive trait from the mother. So we can probably assume that this is an X-linked trait and that this is probably a recessive trait because it's not occurring in the females. So we have to cover two more things, evolution and macromolecules. So evolution is based off the findings of Charles Darwin when he traveled to the Galapagos Islands, and he created the rules for evolution, and he based this off of his idea of natural selection, what we know today as survival of the fittest. Now there are 5 points to natural selection that you have to know. So variation in population. That means that I could be born with 3 eyes and you could be born with 2 eyes. We're both the same population, but we have a variation in how we appear. So some variations are favorable. My 3-eye variation could make me see more than you. So I am able to see more, and that makes my trait favorable. More offspring are produced than can survive. So you might have a mother, she might birth 5 of something, but maybe only 4 of those survive. Those organisms that can survive contain favorable traits. So if I survive, then my 3 eyes must make me have a superior trait or a favorable trait, and that helps me survive. And then populations will change over time. Maybe because I have 3 eyes and because I'm able to survive, then everybody in my population is now going to have 3 eyes so that they can survive until 4 eyes comes along, and that helps them survive more. However, evolution also involves determining the lineage of animals. So we can either use DNA sequencing or amino acid sequencing to determine if 2 organisms are related, but we can also look at their structures. So a homologous structure is a structure that looks the same but serves a different purpose, such as a human forearm but a bat wing. They look the same, but I don't use my forearms to fly, unfortunately. Now, an analogous structure is a structure that serves the same purpose, but it looks different. So think of how maybe a fly will fly, their wings, versus bat wings. They both seem to serve the same purpose, but they're going to look different. And then we also have vestigial structures, and those are structures that are going to look the same, but they're not going to be used within one population, such as like our appendix. And lastly, let's cover macromolecules. So there are 4 macromolecules: carbohydrates, proteins, lipids, and DNA, but we already talked about that last one, DNA, so we don't have to talk about it here. So a carbohydrate is a general term to describe any organic compound that has a ratio of 2 to 1 for hydrogen to oxygen, but that's really sciencey, so just know that carbs are energy. Now the monomers or monosaccharides, or single versions of carbs, are glucose, fructose, and sucrose, and we know these as sugars. Now when there are 2 monomers put together, it's called a disaccharide, and when there are 3 or more, it's called a polysaccharide. So a polysaccharide could be like fiber or something like that, whereas a disaccharide is going to be lactose. Now lipids are another organic compound, and they're going to be made up of the monomer fatty acids, and the polymers for lipids are fats or waxes or oils. And lastly, we have protein, and they're a monomer. Their monomer is amino acids, and we already kind of covered amino acids when we talked about DNA replication in the ribosomes, and we find these in muscles like these. I'm Colin Fogarty, and good luck on your biology SOL. I'm out.